Cells are the smallest units of life. They are classified into prokaryotic and eukaryotic cells. Each component of a cell has a specific function that is vital for its survival. The nucleus is the control center of the cell, which instructs the endoplasmic reticulum to make and transport proteins via the Golgi apparatus. The surrounding plasma membrane regulates materials entering and leaving the cell, while the cytoskeleton functions as an infrastructure for support. These cells also contain important structures such as the mitochondria and lysosomes that contribute to cell survival. The various structures of the cells are not visible to the naked eye even under high magnification. Thus, scientists have often used dyes which stain a particular organelle or structure to visualize them. In part 1 of this series, we will be running an endocytosis assay using lucifer yellow, a popular fluorescent compound used for determining cellular architecture. Here are the objectives. Hands-on experience with the inverted microscope, how to visualize endocytosis, and how to visualize the organelles and cytoskeleton. These are the reagents and equipment needed. Do check if you have everything before you start. Firstly, place 1 ml of each yeast culture in an Eppendorf tube. Next, puncture the lid of a tube with a large gauge needle several times carefully. The TAs will be helping you with this step. Then, spin down the cells at 3000 RPM for a minute. Make sure the centrifuge is balanced before proceeding. Remove the supernatant from the tubes. Be careful to leave the cell pellet intact. Following this, resuspend the cell pellet in 90 microliters of fresh YPUAD. After resuspension, add 10 microliters of lucifer yellow dye. This will be aliquoted for you by the TAs. Then, mix the sample by flicking the tube several times. Incubate both samples at room temperature for an hour, flicking them occasionally to aerate the samples. Subsequently, add 1 ml ice-cold wash buffer and immediately place the samples in ice. Once again, spin down the cells at 3000 RPM for 1 minute. Next, remove the supernatant and resuspend the cell pellet in 1 ml of ice-cold wash buffer. Repeat these steps 3 times. Thereafter, resuspend the cell pellet in 20 microliters of ice-cold wash buffer and leave in ice. Following this, transfer 1 to 2 microliters of each cell suspension onto a microscope slide. Place a cover slip over the cell suspension carefully. Make sure there is ample space for both samples on the slide. Then, observe the cells with high magnification using all, as well as in 100 times all immersion objective. Flip the glass slide upside down when placing it on the lens. Take images of the cells using the FITC filter and with the tungsten lamp filter. These are the images you should be able to see. Thank you for watching. Have a good laboratory session!